Sit back. Thanks. Thank you. Hi, Lori.
Hey, good evening, everybody. Thanks for joining us. Today's Wednesday. You guys know what time it is, 7 o'clock, and it's time for our midweek Bible study and prayer. We have something special for you tonight, especially if you've been kind of watching things that are happening on the news. And some of that is that you might have heard about some of the advances that have come out, news about vaccine, and we've just been hearing different people kind of saying different things and being confused about it. We're going to address that a little bit later tonight, and you might think, Glenn, when did you become an expert on that? Well, I'm not. So we brought somebody else in to talk about it. And but I just wanted to let you guys know, hey, tonight we're still also going to try to have an opportunity to pray for the things that are of concern to you. We want to partner with you. God knows you, and God hears every prayer you pray. But sometimes we just need that power of agreement. And so if you have a prayer request, please text us in at 808-793-5655. And then we also want to remember who we pray to, right? That it's not the fact that if you pray sometimes. It's like, they'll say, the old saying is that even there's no atheists in foxholes. But the issue is this: who do we pray to? We need to remember who it is that we pray to. And one of those things that helps me to remember that is worship. Worship always reminds me of that. And so we're going to have an opportunity to do that this evening before we get into our presentation. And so, hey, let's worship together. And as we do, would you sing heartily unto God, because He's worthy of all our praise. So let's turn our hearts toward Him and worship together.
People come together, strangers, neighbors, our blood is one. Children of generations, of every nation, of the kingdom come. So don't let your heart be troubled. Hold your head up high, don't fear no evil. Fix your eyes on this one truth, God is madly in love with you. So take courage, hold on, be strong. Remember where help comes from. Hold your head up high, don't fear no evil. Fix your eyes on this one truth, that is madly in love with you. So take courage, hold on, be strong. Remember where our help comes from. All you heaven, let the praise go up as the walls come down. All creation, everything with breath, repeat the sound. All His children, clean hands.
your heart's good face, good God. His name is Jesus. Sing wide, all you heavens, let the praise go up as the walls come down. All creation, everything with breath, repeat the sound. All His children, clean hands, pure hearts, good grace, good God. His name is Jesus. Sing wide, all you heavens, let the praise go up as the walls come down. All creation, everything with breath, repeat the sound. All His children, clean hands, pure hearts, good face, good God. His name is Jesus. Jesus, our redemption, our salvation is in His blood. Jesus, light of heaven, friend forever, His kingdom come. Yes, if you believe that, would you say yes, Lord?
Yes, yes, Lord. Hey, good evening again. Isn't He? He is. He's in control. He's good. He's moving. And you know, when we're talking about have courage, be strong, have faith, know where our help comes from. We know our help is in the Lord. And yet, at the same time, there are things that are happening around us that God will do and will use for our good, for the good. And we want to have an opportunity tonight to help you and to talk about some of those things. But again, if you have a prayer request, let us know, 808-793-5655. But we wanted to bring in an expert or somebody who's worked in the field of pharmaceuticals, of development, of treatments and things on the medication side, and the things that, to be honest, I don't know about you, but it's kind of a far outside of my wheelhouse. And so you have seen him before, you've heard him before, but tonight speaking and doing a special presentation is Dr. Clarence Yee. And so some of you might be wondering what kind of Dr. Clarence is. Is he a doctor of theology? No, he's working on his MDiv or Masters of Divinity, but he has practiced medicine for something like 11 or 12 years. I can't remember what it is. He'll tell you. But I was talking to one of our good friends. Some of you guys know Dr. Randy Furushima, and we were talking once about Clarence, and because Clarence had mentioned to me that he knew Dr. Randy, so I asked, "Hey, Randy, do you know Clarence?" He goes, "Yeah. He was my doctor." Like, I went to him for I forget however many years, and so that's how we met. He says, but he did practice medicine, but he has some other things that he's worked on. He has his Masters of Business Administration, and then he went on and did a doctorate in health sciences. And I think he can, if he needs to, he can give a little bit more on that. He's also worked with one of the largest pharmaceutical companies that is out there as a director of science. But again, I might have got some of that a little, that might need a little shaping. If so, he can give us that. But part of that is this whole talk about vaccine, and some people are nervous and afraid. Is that, you know, and there are so many different things. Is the vaccine the cure for everything, or is it something that we ought to reject, or whatever it is? So we wanted to bring a little bit of sane discussion to that from somebody who's been in the field, who can understand some of the things that have been happening, and maybe you might find that you'll take strength and be encouraged, be strong, because God's working, and sometimes we just need to see how He's working. So do me a favor, give a warm welcome to, you know him, Dr. Clarence Yee, and he's going to share with us this evening a special update, special presentation on some of these changes.
Oh, sweet.
have an audience at an N of 3. That doesn't mean that's statistically significant, but that does make me feel better. I'm going to take this off. A couple of reasons why we decided to do this is, number one is because we stopped the Pentateuch because we finished, and then we had no other subject matter, so we didn't know what else to talk about. The other reasons why was, this is actually probably the most revolutionary in terms of technology that you folks are going to see in the way that pharmaceutical medications have been brought about in this last 9 months. And that's unprecedented. I mean, this is talking about revolutionary in terms of being able to find this type of technology. The technology has actually been there now for the last 10 years, but what is surprising is that they were able to bring all of this up, compress it, and make it on parallel trials in just a matter of months. And I'm going to explain those reasons why. Background. So I did practice in internal medicine, and yes, Pastor Randy humbly was my patient. I practiced for about 17 to 18 years in internal medicine. I also did 4 years of emergency room and urgent care, and then 2 years of a dermatology fellowship. After that, I was approached by this pharmaceutical company is probably the second globally, and I was working with them now for the last 18 years in the areas of research. So this has actually become my passion next to God now. Well, actually, no, God first, and then this is next. No, God first, my wife, and then this is next, or my children. So it's something like that, okay? Because she's watching here, and I just want to let you know before we get started, she and I will be celebrating our anniversary. And as you folks know, we both had lost our spouses many years ago. And through tragedy, God brought two people together. And I think taught us how to love God better. And so we're actually going to be celebrating our 10th anniversary in 2 days.
Wow.
So, yeah, I made the contract with her when I first met that it was going to be at least 10 years. So I made that contract, so I'm kind of free and void. I don't know how I can make another 10-year contract, but I think that's what we're supposed to try and do, okay? The people that I brought, or I asked to come today are also, we have a lot of great healthcare providers within this church, and we need to utilize this body in the corpus of church because God made us for so many great reasons. And so I asked, I'm going to use their first names because I think if you later have questions, I'd like you to reach out to them. And that's going to be Lisa and Edna. I forgot to ask Edna, but kind of volunteered anyway, so sorry, Edna. But again, I'll be here as well. And this is, again, as the data becomes vetted and as it finally gets published in the New England Journal, that's when everything is going to be completely out there. And the rubber hits the road. And then at that point, I can define the study much better for you. But I'm going to start with this first. And I'm going to put a laser pointer today, so I'm hoping you guys can see this laser pointer. 1 second. Because I just learned how to do this with my friend, so I'm going to have to do that. Did it come on? Yeah. Can you see it?
Yeah.
Okay, so this is just a real quick first review about what the coronavirus was. Remember, the coronavirus comes from the family of Coronaviridae. There actually have been 7 coronaviruses. The coronaviruses have existed actually for about 2.5 decades, so about 25 years when it moved from zoonotic to humans. So zoonosis means all of the other animals, right? And we know now that this probably had somehow mutated over the last 25 to 30 years from the bats, civets, camels, and now we know the pangolin, which is an anteater. The pangolin and the bat seem to have the most common homology, meaning the same consistency with the genetic sequence as the human style. So the coronavirus eventually mutated over 30, 40, 50 years, eventually then attacked man about 25 years ago. But because the changes are subtle each time, each one of those first 4 coronaviridae actually only caused very mild illnesses. And so it never hit the radar. We never thought about it. Then all of a sudden when we got the SARS-CoV-1, when that came out, and then when the Mediterranean Eastern respiratory disease came out, all of a sudden we got a little more excited and we started saying, okay, now we need to prompt this up. But somehow we were still able to hit those challenges and be able to kind of decrease these effects. And so because of that, we didn't do what we should have done, and that was our homework at that time. Now, when the coronavirus came out, this coronavirus belongs to what we call the beta coronavirus. And I want you to understand that, because when you start reading these journal articles, you're going to sit there and say, I don't understand beta. So basically, they're broken down into 4 subunits: alpha, beta, gamma, delta. Alpha beta belongs to mammalian cells, which is us. Gamma delta is what we see actually in the birds, the avians. So anytime they speak to you about research in the gamma delta, that's going to be preclinical studies looking at animal trials and seeing how well that works. When they talk about alpha beta coronavirus, they're talking about our coronavirus, SARS-CoV-2. Okay? Now, when you look at the SARS-CoV-2, as you remember, the last time when we did this talk, which was it 9 months ago?
Yes. Before that.
Wow, you got a great memory, Edna. Thank you. I'm getting a little bit of Alzheimer's nowadays. So Edna is correct. It was about 9 months ago. Now, what the coronavirus basically does is this. It has basically made up of 4 major proteins. These proteins we know as the top is called the M protein, and all it is a form of amino acid, which is an antigen that sits right here at the envelope of the virus. Okay, and the virus is basically circular. And then this little M protein is, if you can look at this electron microscope of this animation, yellow, I believe, are the M's. The ones that I'm colorblind, so I don't know if this is pink or white or white. White, Mom? Okay. So my wife said it's white. So that's the E protein. But the one that we're really interested in, and just to make it go further so you understand, this is the envelope. The envelope is really made out of just phospholipids. And as you remember the title, lipids, what is that? They're basically made out of cholesterol esters and fatty lipids. That's all it really is. So that's how come it's able to transmit and get into the human cell so easily. But first, it needs a key and a lock process. So as you remember, the human cell has all ubiquitous. And this is called the ACE2 receptor. And it looks exactly like this. There's a lock and a key. The only one that can open this has got to be a very specific receptor binding domain, a very special key. And this is actually found off of these spike proteins. And that's what you're going to hear in the next, probably the next 4 or 5 years. Every single drug, whether it's a monoclonal antibody, recombinant, viral vector, DNA plasmid, messenger RNA, no matter what you hear, all you got to really understand is if they're saying their target is the spike protein, it's right here at the receptor binding domain of every single one of these areas, because that's how it enters the cell. The last thing I'm going to do before I switch this over is understand again, remember the disease state, and this is what I was sharing with Lisa, and they're both RNs, so again, I think they can help us, and they can also tell you about how this injection is going to be in terms of what are the difference between an IM, sub-Q, intravenous, the dosing style. They can even tell you how you have to make sure that, because this thing has to be very ultra refrigerated in terms of making sure you get there on time when you get the injections. But in any event, when we go back and we talk about this coronavirus, what was I saying, Mom?
Lock and key.
Huh?
Lock and key.
Okay, so the lock and key, and when we look at the disease state, is this. It attacks because it's ubiquitous. When you look at this ACE2 receptor, when I use the word ubiquitous, basically it means it is almost in every human tissue. And that's the scary part, and that's why this disease has been so scary when it hits the immunocompromised, when it hits the elderly like me, when it hits the obese or the diabetics. What it does is essentially can shut down a human system. So it starts off in the lung tissue, it starts off in the alveolar branches, which is at the terminal end of the lungs, and also at the small bronchioles. That's where it enters. But then from there it can get into the blood, and then it affects the heart, it affects the renal area, the kidneys, it can also affect the GI tract. We have a very dear friend of ours in the church who had come down with this COVID. I had a chance to see him the other day, and he's really looking good. But what was interesting is one of his major and most prominent symptoms was, yes, he did develop the pulmonary symptoms later, but he actually began to develop first the GI symptoms. So these are the things that you have to understand when we look at medicine and science. It's elegant. What God created, I'm telling you, is elegant. Now, I have to make sure that you see this as a disclosure. And the main thing behind this is with my, can you see my red blazer now? Pretty cool, huh? Okay, no financial disclosures. That basically means that New Hope Kapolei is not paying me, but no one else is paying me. So no paying me. I'm doing this simply because I think this is first for educational purposes, and it's for New Hope Kapolei to really talk to you about these two new COVID-2 vaccine trials. I think they are revolutionary. And I want to make sure that you understand the science and the accurate data, because another person in the church has sent me several videos of these YouTube from people who believe that these are nanobots that are going to enter into our body and somehow be able to take over our intelligence. Some of them is about monitoring our entire systems, and that's really not any of that. And so that's what I wanted to be able to let you know. So we're going to move here from here. Potency of vaccinations. This is just a little bit of history. And the reasons why is we always want to know, well, what does it mean when you always hear in the news, you hear this thing called herd immunity. And it's really pretty easy to understand. It basically means if we cover the majority of people with vaccinations, we protect those people who can't take it. And who are those who can't? This is not going to be indicated for those children 12 and under. So a child who's 6 or 7, pediatric, they could theoretically still get COVID. Your grandson, your granddaughter, any one of our church members. But what it will do is if we've entirely gotten enough herd protection, theoretically the children, and then also those who may be severely immunocompromised, those two categories of people who may not get the drug, those are the ones that we can protect. And so I urge you, after you hear this trial, to really look at the data. It's extremely safe. It's pretty amazing in terms of this messenger RNA technology. When we look at this, so what does it mean in real life? Smallpox was eradicated in 1980. In order for them to eradicate this, they had to actually do an 80% herd immunization, so vaccinate the global world. 80% had to have been vaccinated to be able to eradicate smallpox. And to this date, it seems like we've done it. Now, once in a while, you'll see small outbreaks, but for the most part, smallpox is no longer heard of. It's not a deadly disease. We see measles. That's well controlled with the MMR. 91 to 94% has to be herd immune. The polio virus, 80 to 85%. It is almost eradicated. There's only a few areas of outbreaks in certain areas of countries. But when you look at the SARS-CoV-2, this is determined on what we call the R0, meaning how many people can get infected from one person. So the R0 in the initial trial, we found out was 1 to 2, 1 to 2.5. That later got updated to 1 to 3, meaning for every one person who's infected, they can spread it to 3. I actually believe the number might be higher. But when you look at that, they did the mathematical equations. Dr. Kwok, over here in the Journal of Infectious Disease, he essentially did the mathematical equations and said this, that 67% of the world population, if they are vaccinated, and I spelled vaccinated wrong, so that was a test for you folks. But essentially 67%, so if we can get 2/3 of a global population herd immune, we could essentially control this disease tremendously. If not, maybe even, I could say, in one sense, eradicate, but I don't want to because I know that that's not true. But you could control these. What does that mean in terms of sheer numbers? That sheer number comes down to this. Currently in the United States, we have about 11.4 million cases, 248,000 deaths. Globally, we now have about 55.6 million cases. 36 million have resolved, but we have about 1.36 million deaths. That is what we're trying to prevent. So when we look at this COVID vaccine, we have to actually, looking at 67% of the population, we have to come out with 5.3 billion doses. That's crazy phenomenal. That's why this mRNA technology was built, this platform technology. It needed to be done quick, it needed to be done safe, it needed to be done effective. And that's why I'm pretty impressed by what Pfizer and BioNTech has done, as well as Moderna. CureVac is also producing theirs, and it should be coming out maybe in 6 or 7 months. And there are other modalities. So when we look at this portion, I'm going to switch gears to you and say, well, why don't we use the traditional vaccine development? The number one reasons why is because this is what I was involved with all these years. Not only vaccines, but cancer research, liver diseases, rheumatology diseases, cardiology, neurology, multiple sclerosis. I mean, it was on and ad infinitum, ad nauseum. But the traditional style of research takes time. And we didn't have that time. So one of the things when I say time is just to produce this vaccine, the traditional style, you first have to grow the virus or grow the bacteria in culture plates, and then you have to grow millions of this, actually closer to billions of this, right? And then you have to make sure that all of these stay alive during the time that you incubate and you grow this. So you have to have a size way bigger than this ministry lead center. Pretty big, okay? And then you have to make sure that these things are all fed properly. Because again, if you give too many nutrients, it dies. You don't give enough nutrients, it dies. You give enough oxygen or not enough oxygen, it dies. So all of these things take time. Once this is finished, then you have to then remove the virus or the bacteria, and you then have to isolate this. From here, you then have to purify this, because in this batch, there are still areas of embryo egg or cell culture medium, and you have to essentially get all of that out, because if that is placed in a vaccination, that creates problems of contamination and other disease enhancement, so that the patient could actually die. So now this takes another extra process of ultra, and all of this takes time. When they finish with this, they finally go in and they say with certain adjuvant therapies, this is the type where you have to initiate the immune system to kick in. And essentially you place this adjuvant in here, and then you give the injection, and then the people get this. Okay? Now, this then, once it's completed, these are the large bioreactors and fermenters that you've got to keep this system alive all this time until all of the injections are given out. Then you've got to make sure that they're placed in these in the vials for the injections. They have to be labeled. Everything has to be FDA approved, and then you have to make sure that the distribution center is set. So all of this time, from the time you start that to the time you get this out, usually takes 10 to 15 years. And that's what we didn't have time for, right? I mean, can you imagine wearing a mask for this long? I don't think we could. The global economy couldn't handle this, right? And if you really look at this, even though despite we are wearing masks and we are probably reducing some of the risk, we're still seeing outbreaks. And you're going to see this even in the state of Hawaii. You're never going to see coronavirus go down to 1 or 2 cases or 5 cases a day, just simply with the use of the mask and distancing. It's too difficult. So we know that the vaccine studies are the best way to hit the first front end. Now, how did we do this? This is where I have to at least say the present administration, you have to give credit. They put tons of money, because that's what you need first. You see, anytime I have to do a drug trial, I have to go to Pastor Glenn and say, okay, I want to do a trial on drug X, Y, and Z because I think it's going to make you and I taller. And you go, oh, I like that idea. Yeah, it's going to make us really much taller. But I need $1 billion up front, because it's going to take 10 years for me to get it through the FDA, to write all of these protocol summaries, to go from preclinical, because I try it in a monkey first, or a rat, or a rabbit, and then I eventually get into human trials, and then I move it in. And so we have all of these different studies from phase 1, where you're going to see some YouTube people were claiming all these severe side effects on a phase 1 trial, but again, statistically, they were doing it incorrectly, because a phase 1 trial is usually less than 100, a handful of people, just 40, maybe 30 healthy people. So even if 1 or 2 people get side effects, that percentage looks huge. It looks like 20, 30% got the side effect. But the reality, that's not true. So we do these phase 1 trials, we send it to the FDA, they take 6 months to look at it, then they tell me if they like it or they don't. What are the amendments I have to change to the protocol to phase 2? I move to phase 2 and I get a bigger group of people now 250, and some of them with a disease state. I treat them, I get the results, I write up the results, I send them back to, again, FDA. They look at it again for another 6 months, and then they come back and say, I don't like this, I like this, I don't like this. I make more amendments to the second protocol, and then I send it back to them again. And then finally, when we get into the phase 3 trials, almost 7 years has gone by. And that's the insanity of what we have to do. It is interesting that this present administration wiped that entire slate clean. And what they did, and when I show you these trials, they did not fudge on safety. Okay? And that's really important, because you are going to hear time and again in the news, hey, but did they fudge on the safety? Did they fudge on the safety? And I'm going to tell you, these two drug companies won't do that. Now, Moderna is brand new, but just because it's brand new, it doesn't mean it's brand new. Those researchers are probably the best in the world. I'm going to move quickly over here next to this. Operation Warp Speed, we're going to switch, but basically you need these types of technology, a platform technology, an accelerated program, and now you know you need the money. Okay? This is just kind of showing you all the different trials that are now going on, plus 200 more. So I think we're getting now to the point where we are actually going to sit there and turn this corner probably in the next 6 months. When we look at this, I'm going to quickly move over from the ACIP data because I know we're short of time. Okay, so over 200 COVID-19 vaccine trials. Within the United States, we're going to be looking at the 4 vaccine phase 3 clinical trials. I'm actually only going to specifically talk about the 2 phase 3 vaccine trials that have recently been released. And then that's the one by Pfizer BioNTech. And then the second one, again, is by Moderna messenger RNA 1273. Okay? So these vaccine trials are here. What I really want you to focus on is this, the sheer numbers of the patients. I mean, we're dealing right here, just alone with the 4 trials, that looks like, what is that, 120, 160,000 patients. That amount of data, when it comes back and it says there are no serious safety side effects, that's statistically significant. That's powerful. So when you have somebody on YouTube saying, you know, their phase 1 trial had 45 patients and they had 50% who complained of itch and burning rash and pain and fever, you're going to see a very different number when you look at this data. And that's really the important thing, because you want to be able to tell yourself, do I want to take this injection? Do I want to protect myself, or am I going to get into some kind of a major side effect? I don't know if you can see this, but these are the different methods of the studies. And I know because we don't have time, I'm not going to go into these. If you want to know more of them, pull Edna aside, or myself, or Lisa, and we can talk story. But this is really looking at recombinant viral vector-based diseases. And they're going to look at this as a viral vector that's going to introduce. And remember I told you when you first saw the animation, what do you have to really know? This is just a little animation of what we have to know every day. And I could show you slides that makes your head spin. But this simple slide really is going to talk about only one thing, the S-spiked protein. And then now they're going to show you just different methods of how the intracellular biology is able to convert a protein to protect our bodies, an antigen-antibody reaction, okay, to protect our body against the spiked protein. But you first have to get the spike protein in the body and then into the cell, and then you have to transmit this and make this into an antibody. So you have a virus vector that can do it, or you can do it by a DNA plasmid. The problem with the DNA plasmid was it takes a 2-step process, but I don't think that's going to change much at all. Some people were saying when this first came out that this was going to cause a transcription of our human DNA and cause mutagenesis. That's incorrect theory. And the reason why is because a DNA plasmid does not go into the nucleus. When it hits the cytosol, it breaks down into messenger RNA and it's translated, but not transcribed. Different, okay? And again, if you need further verification and understanding, I'm going to depend on my colleagues. I'll be happy to help as well, okay? But this is the one that we're really looking at. Messenger RNA vaccine, right here. And this is the puppy that is so amazing, because what it's using is just one piece of machinery in the intracellular biology called a ribosome. And what the ribosome does is it makes messenger RNA. It makes protein for messenger RNA. So this messenger RNA is just a sequence of the spike protein. Very simple. Okay, I can explain it more technical, but very simple. The spike protein. That then is what they want to get into the system. So instead of growing the spike protein, which could take them 2 or 3 years to batch it and do everything else, and then get FDA approval, which would take another year, what they're going to do is allow our own human system to make the spike protein ourselves. And so it goes through these transduction signaling pathways. It eventually hits this thing called an endoplasmic reticulum, which then eventually says, I'm going to connect you to 2 devices that's going to improve your body's ability to make antibodies. And what we need is this thing called the adaptive and the innate immunity. Innate means we needed to just immediately gobble it up. And from the ladies' days, you remember macrophages in the lungs, antigen-presenting cells, they eat anything. So you need that first system to work. And then the second system you need to work is an adaptive immunity, which develops the antibodies, and then develops this thing called cytotoxic T8 cells, or CD8. And if you can see, I don't know if my picture is big enough, but this is the major histocompatibility of CD8, which is MHC1. This is MHC2, which does the CD4 T helper, which drives the B cells to make a memory. And you want a memory, just like in the in our computers and the recent RAMs, they call it. If I want to say, hey, gee, I remember reading that trial, I just type in my history, COVID, and then all of a sudden, boom, everything comes out instantaneously. That is what the memory cell does in the human biology. You have to make sure you have enough B cell memory, because when you get infected with this disease, you don't know when. It could be 3 days if Lisa goes into the ER or the surgery center, or it could be 3 months, right? So you want a memory cell that's going to hold persistence over time. This is Moderna's messenger RNA mechanism of action. It's the same picture, but just 2. What you're going to look at is if I circle right here, that's the major histocompatibility 1, and that's making your CD8 T lymphocyte. This is the major histocompatibility 2. That's making your TH4 helper cell, which drives to the B cell memory. So now you've got memory, now you've got an attack system, and now you've got innate activity. You've got all 3 parameters covered just by this. And it does it within 7 days after inoculation. And the study showed after 28 days, it seemed to show tremendous protection. So this is Pfizer's. I'm not going to go over it because it's exactly the same thing, just a different animation. MHC1, MHC2, CD8, CD4, B cell memory. Okay? This is how a lipid nanoparticle looks like. It's not made out of a robot. It is not going to control our lives. It's not going to take away our economic systems. All it is are phospholipids. These phospholipids, as you see up here, are just made out of fatty tissue like cholesterol, or these hydrophobic payloads, or this pegylated lipid. All it is oils. So what will happen is this. It's so cool. When it hits that cell membrane, it goes right through. Once it goes through that, all it does is it dissolves through the natural reticuloendothelial systems. The white cells just gobble it up, just like, hey, it's food. They eat it. Then the payload is delivered. Here is this RNA, the messenger RNA, that now is going to tell the endoplasmic reticulum to start making protein. And that protein you want is what? The spike protein, right? The S protein, that little red thing that you saw. You want that receptor binding domain. That is how it gets it into the cell. Now, Pfizer BioNTech, their drug is called BNT162b2, okay? And you just got to remember Pfizer BioNTech, that's the drug you want to look at. And basically in this one real quick trial, I just want to let you know of 3 major things. The word rapid, the word safety, and the word healthy individuals. So when they made these parameters in their protocol summaries, The protocol summaries for Pfizer and BioNTech is 142 pages. So I did read through that. The Moderna's protocol summary is 186 pages, and I read through that. They also ended up with 36 pages of amendments by the FDA, and I looked at that, okay? So I can tell you, you cannot just make this and manufacture it without serious thought of what you're going to do. Now, what they did was this. They did this so rapidly because they had the money. They compressed all these 3 trials into one fast trial. And I'm going to show this to you on the next slide. But when you look at this, the first thing they wanted to examine was not tolerability, not immunogenicity, not efficacy, but safety. That's really key here. Because if you kill a patient, they get sued. We get sued. I remember when I was in the pharma industry, that was our major concern. We have a massive side effect, we're in trouble. So that's why they look at this first. And then it was all given in healthy individuals. This was their study design. It was compressed from phase 1 here. They quickly looked at the 3 different doses that they sat there and said, which one will work the best in a human? Now normally I would have to just do the 1 trial, submit it to the FDA like I talked to you about, talk story with them for 6 months, get all these amendments to the protocol, and then redesign the trial again to a protocol number 2, a phase 2 trial. And then if that was finished, I then would agree. What they did was they did a seamless compressed trial of going first to phase 1. Immediately as they were getting results, they were already recruiting for phase 2 and phase 3. So on the backside, they had recruiters going out and already getting the patients. They were all ready. As soon as this trial came out, this data was already being fed to the FDA 24/7. So instead of them saying, wait for 3 or 6 months and then send me the paper, they were getting everything. Once this came about, they then sat there and says, okay, here's your criteria. Here's the criteria. Multi-centered, multinational, approximately 215 sites, a seamless and continuous phase 1, 2, and 3 trial. Had to be randomized, meaning that computer separates these people into 2 equal groups, comparatively speaking. Placebo controlled, have to have a placebo in order to make sure that the drug really works. It had to be an observer blind, meaning that the person who gave the injection knew what it was, but the person who was actually doing the initial studies and doing all these parameters did not know who got what. And that was important because the person not supposed to know. Now why should the doctor know? Well, it makes common sense. If I give this to Edna and she ends up with a cardiac arrest and I know I gave her the drug, I need to get to her right away. If I gave her a placebo and she got a cardiac arrest, well, then I'm going to look straight for cardiac reasons and not because of the drug. So if that makes sense, kind of, yeah. Then what they did was this. They looked at 3 age demographics. So with Pfizer BioNTech, they were so smart in their designation. They started off in phase 2 from age 18 to 65, one group. Second group, 65 to 85, that's my group. But then as they saw the safety data came out, they moved into the younger category and then went from 12 to 17. And that's important. Total number of patients, almost 44,000. So 43,661 subjects, 42% came from the global participants, 30% were considered, that's wrong spelling too, yeah, Mom. That's wrong, right, racially? I don't think so. Sorry, I'm dyslexic. 30% were U.S. racially and ethnically diverse, meaning we did have Asians, we did have African Americans, we did have Hispanics, we did have Caucasians. And then again, 41 to 45% were in this age category. So you see, they hit all the numbers. Everybody who was supposed to be immunocompromised or at high risk, this is what they were looking at. Then, this is the participant diversity. I don't know if you can see it too well, but basically Asians was about 5%, 5.5 in the U.S. Blacks were about 10, Hispanics 13, my age between 45%. Okay, and again, remember the number was sheer, was large. We're dealing with 44,000 patients, 43,661 to be exact. Okay, but when you do that, 10% of 43,661 is 4,360.1 patient. Okay, so that's a large number of Asians. I don't normally see that kind in this trial. Now, when you add that with the same thing with Moderna, you've got now 9,000 cases. You add that with CureVac, you've got now 13,000 cases. So you see, it all starts to add up. When we look at their analysis, BNT reported a 95% efficacy. Their initial results just reported 4 days prior to this, or I'm sorry, 7 days prior to this, was actually saying about 90% efficacy. They got their final results, and the final results has to be when the confirmed cases was hit at 164. That was reached yesterday, and they vetted the data and released it today. That is insane speed. That means nobody is sleeping at Pfizer. They are working in 3 shifts of 24/7. That's how dedicated these scientists and researchers are. So if I had that, I'd be asking for a lot more money. I just wouldn't do that. Okay, anyways, so 95% efficacy. This p-value is 0.0001. Now, if I were to look to Lisa and I were to look to Edna, or even to Pastor Glenn, they would understand, hey, that sounds pretty statistically significant. Anything that is less than 0.05 is considered statistically significant. By New England Journal of Medicine, once it reaches 0.0001, it doesn't matter if it goes any further. What does it mean? What we have is a null hypothesis first. We all generate a null hypothesis. So the null hypothesis for both of these trials was this: our drug, BioNTech 162b, does not work in the COVID-19 patients. That is their hypothesis that they start off with, that this drug does not work. Now, when it reaches a p-value of statistical significance of this nature, it says you have to reject that thought, because that means that thing doesn't exist, because this is not by random. So meaning the results that you are now going to see, you have to look at this and make your inference. So when we look at this, it's going to say, well, 95% effective, then that means if I do another repeat trial like this, I'm going to get pretty much similar results. Of the 170 confirmed cases, 162 people who came down with the COVID was all placebo. That's the cool part, right? Because now it's telling you out of 21,750, only 8 people came down with the COVID but had been vaccinated. Okay? Now, what I want to see when the real data comes out, though, is how much of these people were exposed to COVID. I mean, were they just allowed to go out in the community? Or did they work as healthcare personnel? Because, you know, that's going to be different, right? If you put somebody in a hospital working 24/7, 7 days a week or 5 days a week, and then they come back and you've got 41,000 of them, and you say, oh, none of them came down except for 8? That is so massively statistically significant, I'd be standing in line for the shot. Okay? Now, the safety data was also looked at 43,000 subjects, and this is the safety analysis. And that's the part you want to see. And I want to be very clear on this. The data is not yet complete. You need to wait till it gets published in the New England Journal, and then when we see all the demographics, exactly what it is, then we can say with a surety the drug is safe. But with that being said, they looked at first adverse events and serious adverse events. And what is the two difference? Adverse events are usually any medical occurrence that can occur once you're giving a device, a medication, anything. I go back in, I have a rash, they put it down. I come in, I said, I think my wife looks pretty. They put it down. If I sit there and say, I have a headache, they put it down. Anything that you complain about after you've had the injection is considered an adverse event. And then they compare it to the placebo. And then at that point, they can sit there and say, anything that is more than 2% is considered a grade 3 adverse event that makes me want to stand up and say, okay, majority of the people who take this shot, they may get this kind of symptom. And so those who were seen was fatigue and headache. Fatigue was seen at about 3.8%. and headache at 2, but not much. Nothing else was reported. It's crazy. Serious adverse event, there were 10 cases. Now, what is serious adverse event? Death, hospitalization. They get the COVID per se. Well, no, that won't work on that one. They would die, or they would get renal failure, or something of adverse event, cardiac event. Okay. Now, This one makes me stand up and look until I separate the data. There were 9 cases that all belonged to the placebo, and only 1 case was in the COVID. Okay, that's statistical. That you can't change out of 43,000 patients. Now, when I look over here, the Data Monitoring Committee then did not report any serious safety concerns. Now, for us to get this as a emergency usage authorization, this is a very specific code. It means this: you do not fudge on safety. Okay, you cannot fudge on safety. But what you have to do this is you got to show me first safety, 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 safety is there. Okay, now after that you got to show me that the efficacy outweighs the benefit or the risk of getting the disease. And that's why you see a 95% effectiveness with less than 2% and only on fatigue and headache and only 1 serious adverse event, which they haven't told me what it was, but it would be interesting to see. Okay, the dose was given at 30 micrograms times 2. And this is just kind of quickly show you schedule of activities and procedures. And this is what I would do to check for protocol violations, meaning the doctor wants to do the trial and I say, okay, fine, I'll allow you to do this. I go through everything. There are usually 10 pages like this on every single one patient. So every single time that patient comes in, those guys have to make sure they hit all these protocol marks for me on 10 pages, and I will come in and I will look at it. If they don't do this, this is a data breach, a data protocol violation. First time is forgiven. Second time is not forgiven. You're only given 1 chance. Second time you get fined. Third time you're kicked out of the trial. So that's why safety was never fudged. And when I looked at the protocol summary of 142 pages, they had 10 pages of these on every single one, showing what you have to do. And they looked at things like informed consent, participant number, demographics, latest CD4 counts, looking at the B cell measurements, vital signs, height, body, etc., etc., etc. It just went on and on and on. So that tells me again, they didn't fudge on the data. They just compressed everything because they got the money. They had the money to do it. I hope this makes sense. Moderna, messenger RNA 1273 vaccine trial. This was the breakdown. And you can see in them, they had 43% were white, 20% Hispanic. Asian population can also mean Filipino, just to let you know. Blacks, 10%. This broke down also into the age category groups. We also went into the gender with Moderna. 53% were male, 40% were female. And then this also went into risk stratification. So Moderna gave out a lot more information. They sat there and they told me 1/3 of these patients were diabetic, 1/4 of these patients were severely obese, probably a little bit like me, I think. And I can't see colorblind, honey. So which one is this color? 19%. Is that COPD?
Significant cardiac disease.
Thank you. So that was significant cardiac disease, and this one must be COPD?
Chronic
Okay,
lymphomas.
COPD. Okay, so they looked at all the high-risk patients, and they weren't afraid to incorporate these types of patient into the trial. That was important in the COVID trial, right? The other thing is they also looked at the age, and this was the biggest thing. 58% of that group was, I can't see
Less
this.
than 18.
Oh, that's the healthy guys, yeah? Oh, I'm sorry, it was quarter. 1/4% of the group was greater than 65. And that's again important because these are the ones who end up in the ICU. So again, if you can protect them, they don't ever go to the ICU, right? And what happens? Medical economics goes down. And if medical economics go down, first you save all of our healthcare providers, and then you save the money, and then the global economy goes back up. So I hope that makes sense. That's my MBA side talking. Sorry. Moderna's then was again, same thing, phase 3 trial. It met its statistical criteria, just like Pfizer. Look at this stuff. I mean, this is crazy. 94.5%. They use the same type of technique, messenger RNA. Look at their p-value. I mean, it's like they cheated everybody. 0.0001. Same. NIH appointed Data Safety Monitoring Board that informed Moderna that the trial now has met the pre-specified statistical criteria. So what am I telling you this? the trial was stopped early. Now, it wasn't stopped like in some trials where we see it stopped at 2/3. The trial was probably stopped at around 4/5 to 5/6, almost at the end, but it still was stopped early because they sat there and they said this pre-specified data that the safety committee is looking at as harbingers of problems, they're saying, hey, there's nothing anymore. So now the amount of efficacy is so far away from the placebo that the two measurements are like this. So now they look at efficacy and they said if it's this far, then we are no longer being ethical to the United States if we say we're going to wait another 3 more months. Why? Because when you looked at the 248,000 deaths, in another 3 months we'll have another 20 or 30,000 deaths. That's why they separate and they said it's met criteria, publish, okay? And then that's my job, or used to be my job, and other people's job to vet the data. So their interim analysis, they had 95 cases, I'm sorry, not 90, but they had 95 cases of side effects here. Of the 95 cases, very similar again to Pfizer, 90 cases, all placebo. Only 5 of the 30,000 patients was from the COVID. That's interesting. Secondary endpoint was 11 severe cases. Of the 11 severe, only 10, all 11 was placebo. Let me make sure. Yes, that's crazy. So all 11 people who had severe cases, none of them were in the COVID study drug. So when we look at safety concerns, nothing. So these were some of the side effects that you might encounter though, because they were greater than 2% in the Moderna trial. I suspect you probably are going to get some of these. Now, these are also the same kind of things that you get whenever you receive a flu shot, right? Yeah, Edna's going exactly. And why? Because when you get a flu shot, you're getting actually a live attenuated vaccine. You're getting actually the true virus, but it's weakened. And because of that, that's how you immunologically develop an antibody to protect yourself. But in doing that, you get mild symptoms of this. So things like fatigue, myalgias are muscle aches, arthralgias are pains in the joints, headache, pain, injection site reactions, and erythema, meaning redness around the injection site. Very common. So again, nothing here that to me, I mean, what do you folks think?
Yeah.
Preliminary, yeah? And again, I want to emphasize it's preliminary, but I don't think they're going to walk away and tell me, clients, that's pretty amazing. It doesn't make sense. It's like they cheated. And I hope they didn't, but it doesn't look like they could, okay? It doesn't look like they could because all of these people are independent committees. Conclusion, you have to do these things at the end, and this is what I'm waiting for when the data gets vetted. I want to see their B cell memory numbers. I want to see that sucker go up. I want to know that you have persistence. That means immunologically, you have the memory cells to produce the antibodies like this, so that when you get a secondary response to the spike protein and you get infected by whomever, then essentially what happens is this should mount a quick response and quickly get rid of the S spike proteins, which means it gets rid of the COVID. You will want to see the T cell numbers. CD4, CD8. You want to be able to see what those look like. And then, of course, all the other data has already been shown in terms of its efficacy, so it's there already. Now, the other thing we want to make sure as we look at is this. The biggest problem here is stability. Because with the Pfizer data is you got to keep this frozen, ultra frozen, and I think Lisa and I was talking about this. You got to keep it under negative 70 degrees Fahrenheit? Centigrade? It's got to be frozen, dry ice. So how are they going to do? Remember I showed you that stuff of the truck and the distribution and the chain supply? That's the biggest weakness. They got to figure out how to get it to us without compromising the integrity of the proteins. Moderna's is a little bit better. It's at -2. Now why the two differences? I don't know. Well, it's a -4 degree Fahrenheit, -20 centigrade. But essentially Moderna's, you can actually just carry it in a refrigerator. You freeze it, that's all you need. But you don't have to ultra freeze like Pfizer. So Pfizer's got a little work to be done, particularly although they say in their news that, hey, we've done this on 43,000 patients, we can do this. But they're gonna have to now do billions of doses. The 1.95 billion that was given to them by the Trump administration and the 1.5 billion that was given to Moderna by that same administration, those guys with that money have got to figure out how to get billions of doses, because the first doses that they signed off with that money is 100 million for each company. They are going to be delivering 200 million. By the end of this year, they're supposed to each deliver 50 million doses. So that 50 million and 50 million means 100 million doses. By December, healthcare providers like my colleagues, whom I deeply respect, they're going to get first chance at it, unless I go back into practice, which I don't think so. I think God's calling me. So what are the applications? So perhaps we really actually need to understand this, that God approaches humanity as a reflection of his image. And remember, we've said this time and again, that God made us in our image, Selem. Selem is a reflection of God. So if we had stayed in the garden, all that is good would have been ours, right? Including this stewardship, and Pastor Glenn taught me this, the dominion over all creatures of the earth, even viruses, okay? But we didn't do it the right way the first time. And because of that, we live in a world that has both the tree of the knowledge of good and we have evil. But one of the things we should be celebrating today, one of the things we should be applauding today, is that these people of science, they should be applauded for the good that they have done. Because you just cannot, it's amazing. It's so exciting. I was telling Laurie, if Moderna called me tomorrow and asked me to be a scientific director for West Coast, I would sign up. But then I remembered I've already given myself to Pastor Glenn in the Master of Divinity program, so then I have to sit there and say, I can't do that. But that's how exciting to me this is. The other thing is this, we cannot forget this. As Christians, at the end of the day, we have to thank our Father, because it's him who brought us these men and women of science. And so before Pastor Glenn prays for all of you folks, and I know that's really important, so I hope you're sticking this in, I just want to say a prayer to you folks for 10 seconds. And so if you can bow your heads, I'm going to read this because I don't have the ability to memorize all of this. Dear Heavenly Father, we thank you for this day, our daily bread, that you provided for us, even during the most difficult times in our lives. We pray for those who have fallen, for those families who have had to endure either illnesses by their loved ones or even in their passing. We are grateful, dear Lord, for all these good men and women that we believe came from our God, who has provided this critical bench research, the healthcare and compassion, and this love and understanding that this world so much needs. So allow these outcomes today and more that will follow. Allow this to renew your spirit. Allow this to touch our hearts for one another and to fill us with peace and joy, because we are really grateful, dear Lord, for this day. In your precious name, amen. Thanks, Pastor Glenn.
Can I ask a question?
Yeah.
Someone texted in, are the three major companies sharing information and working together? If so, what's the difference among their products?
So, messenger RNA technology platform is based on three different types of technology. This is going to get a little technical. So one is called the modified messenger RNA technology, another one is called an amplification method, and another one is called the uridine mRNA method. The one that Pfizer used was a modified messenger RNA technology. They use this because it's a little bit safer, but it can drive effectiveness, and it's very quick. The messenger RNA technology that Moderna did, when I looked at their protocols and I looking for mechanism, redaction. So I don't know if you understand what redaction is, all blacked out with the black magic marker. So some parts they don't want to release because of intellectual property. I think eventually when they hit the market and they start saying in terms of all the conferences, they will release this. And then you can get a better understanding. I think for the most part, the real big thing is the messenger RNA technology has never been done before. That's why people were criticizing it so much. But what they don't really understand is this. I'm going to show you one more slide real quick on how this all began almost 15 years ago by this amazing woman. Did it pop up? Yeah. Okay. Her name is Jennifer Duwadna, and on the one with the black coat is Emmanuel Charpentier. These were the 2 Nobel Co-Prize winners of chemistry. These people started genomic editing and started making it a complete science, because up until then, when we tried to do gene therapy, immunotherapy, and play with the human DNA, we made more mistakes than more positive. And so when we were just about ready to give up, these two and this wonderful molecular biologist in chemistry sat there and says, hey, you know, I've been looking at these prokaryotes, these bacteria, these archaea, and I'm saying, how do they protect themselves? They're being invaded by viruses all the time. I mean, they get invaded by viruses more than we do. And then she found out that there was this editing system in there called this. And this is called the CRISPR-Cas9 enzyme system. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. So if you can repeat that, that's great. If you can't, just call it CRISPR. But essentially, she invented this, and she put the scissors here and the scissors here with the Cas9, so that now it cuts the exact portion of the broken DNA that's causing cancer, and it removes this. And then it completes it by correcting the DNA system. This set the seminal papers for ribosome therapy, for DNA therapy, for gene therapy. And that's the major difference. Why do I key on Jennifer Duwadna? She actually is from Hilo, Hawaii. So at the age of 7, while playing out in the fields of Hilo and looking at all the butterflies and all of these other creatures, she was saying, this is what's going on here. And she figured this out. And I think it's amazing. So, long story short, I think all of the messenger RNA platform technologies are going to be somewhat similar, with the exception of how they generate the protein. At this point, looking at the sheer numbers at 30,000 and 43,000, I would be surprised if CureVac, who's coming out with their 3rd messenger RNA technology, with a number like 82%, unless they've done something really bad. I imagine they're going to hit 90% plus, okay? The other technology, like the DNA plasmids, I agree. Don't know yet. We'll have to wait till the trial comes out. It's just really into early phase 2, phase 3 trials. So it also is going to make the spike protein, which supposedly will trigger MHC1 and MHC2. But one thing that I've learned in medicine is, I showed you just one little pathway. There's actually within that cell hundreds and thousands of redundant compensatory pathways. So when one turns one off, it may turn off 5 or 6. So I don't know whether the DNA plasmid technology, although it has been around longer, whether or not when it's going to affect the area of coronavirus, if it's going to get the same kind of efficacy percentages. So that's a great question. The viral vector one, the only major concern I get with this, again, is if you are introducing a foreign antigen, there is a higher risk for maybe disease enhancement, meaning that you are giving a vaccine, just like when we give the live attenuated vaccines. Some people actually really get sick, not just mild myalgias, but they actually get the disease because we introduce the disease. So the viral vector may have some component of that. Again, I'm going to be very cautious and say, but we don't know, because again, no data. So it could be just as good, or it could be a little bit less. So that's where now we have to wait. But the exciting part is it's telling me this. We got 2 mRNAs with positive, statistically significance with huge numbers. Unless CureVac really just messes up their trial, I imagine they're going to come out with similar studies. And now you've got 3 companies that are going to be producing 100 million, 100 million, 100 million. So if you get my drift from a global economic pathway, they're going to be building 1 billion, 1 billion, 1 billion. Within a matter of a year, you've already got 3.5 billion doses. You see, now we're reaching towards herd immunity and decimating the disease. Does that help? So, I'm going to go.
Yeah, no problem.
Thank you.
Thanks, Clarence. Thanks, Doctor. Hey, so I'd like you all to take out a sheet of paper, blank sheet of paper and a pencil, and we're going to have the pop quiz right now. No, I was kidding. That's what kind of had that feeling, you know, we're just like sitting in a class. But I hope you understand that, you know, that we're taking some time where we normally have a study, but that at times, to be honest, we pray often for peace. We pray for God to move in the bodies of our friends and our family or our own. We pray for these kinds of things often. We will pray for God to change circumstance. And God is answering. God is always hearing. God is always moving. And sometimes He touches it apart from the hand of man. And sometimes He touches it as He touches the hand of man and becomes that agent for God. And so here's the good news: be encouraged, be strong. Hey, at one point, you know, when these therapies and vaccines are delivered, and we're probably not going to have an option to say I want Moderna versus I want Pfizer. It's going to be what comes into our arena. And so, you know, because they're going to move these things out by geography and all that. So, we understood the question. Hey, I just wanted to thank God for a couple things. There's some folks, our friends out in Nanakuli, who've been racked with pain basically for a number of days, and then we prayed, and guess what? God answered, and He said, hey, couple pain-free days. And there just was constant cramping and trying to balance this thing from, you know, his condition. And so God is the great physician. So He's hearing prayer. He's moving in situations like this. But we also want to pray. There's I'd like to pray for some friends whose mom went home to be with the Lord today. She fought the good fight. She loved Jesus, walked with Him for many years, and to her it's homecoming. To my friend and Nishida family, it's bittersweet, right? Because we know where mom went, but we also grieve, right? Still humans, and so we just want to pray for that. We want to pray for you just as we close. Father, tonight we come as the family of God, as sons and daughters, and we understand that we are grateful that it is precious in your sight when one of your children goes home to be with you. Because to them it is homecoming, to you it is homecoming. But we are thankful, God, that you know on this side of eternity we experience grief and mourning. And we pray for a covering over that Nishida family. And pray, Lord, that each one would know your goodness and know your kindness and know your comfort. That each one would experience, Lord, those who are weak and those who are strong. Those who are receptive, and to those who have been hardened by some of the circumstances of life. We thank you, God, that you care for them. And Lord, we are praying for our friends who have been watching and joining us today, that Lord, cover them today. And Father, I pray that we would not be so coward and at home that we cannot take the steps of faith that you call us to. Because Lord, you are at work. Lord, we are not saying to be reckless, but we're saying be strong and take courage, for our God is with us in every area, in every step. So Lord, we receive that. Pray your blessing over every person and their family, and God bless you. Hey, thanks for joining us tonight. We'll be here Saturday for 8:30 to 9:30 for prayer and healing. Also, just a reminder that we're having food pantry this weekend as well. So that's our regular food distribution. And a number of exciting things happening on the weekend as well. But have a great day. Have a wonderful rest of the week. We hope to see you guys Saturday or Sunday sometime this weekend. Take care.
Aw.
Is it okay?
Oh, in living colors. I love it. It's like a coronavirus thing. It's in living colors. She did it presentable. Oh,
Are we off yet?
oh.